Alrighty, today we are going to be talking about a number of things, that including and starting with skeletal muscle. So, right here you can see that I've drawn out a couple little things just to help this video go a little bit faster, but also to just make it so that these drawings will be a little bit more clear because usually when I make them beforehand, they're a little rushed. So, I did this just to try to make this video a little better, see how it works. Right here, what I'll start by labeling is something. So imagine right here in this region, we have a tendon connection. And then if this was a more large scale, like chunk of muscle, this would kind of be where the muscle belly is. And then if you were to take a cross segment, like I kind of drawn here, if you were to chop this in half, you would see these little, like ring-like structures like I've drawn right here. And these little ring-like structures have something around them called the perimysium. And the perimysium really is just a little dividing membrane in between all of the smaller, intricate fibers that you kind of see drawn around, or you kind of seen drawn like as these little circles right here. And you have a bunch of like nerve innervations. So you'll have like, let's do a little like line. Like let's say you got nerve innervations, you get blood innervation through here, like through vessels and veins. Anyway, these are going to kind of run on this outside like circle of the perimysium is where you're going to find these little nerve and blood and vessel not innervations. Innervations obviously just for nerve, but like the vasculature. And then this little area, like this whole chunk right here that I have blown up. So like going from here out. So in this, you're going to see a bunch of little tiny rings, and that's what I have drawn in this area right here. And so this outer layer, or this section right here, is what we call a fascicle. And a fascicle really is just a smaller division of the whole muscle itself. Then we go inside here, inside the fascicle, again, we have more subunits. So what you'll see is this is kind of just like, there's like smaller parts of similar looking structures as you get deeper and deeper into the muscle cell, which we call our sarcomere, by the way, and I'll get into that. So this little blown up thing right here, which is what I've drawn right here, is what we'd call an actual muscle fiber. And inside the muscle fiber, this is really where the magic starts to happen. Because inside each of these, on these little layers, like if you were to blow one of these up, you would kind of have like this structure where you would see this little like yellow membrane that goes around it. And that is, that's actually our sarcoplasmic reticulum. And that's where, like I said earlier, the magic's really going to happen. That's where we're going to have the calcium exchange and such to start creating an actual muscle contraction. But we're going to go even a little bit farther than the actual muscle fiber itself. We're actually going to go all the way down to these individual fibers that you would find inside of this muscle fiber. And that's what we call a myo again meaning muscle, fibro. Myofibril. And this myofibril is where we're going to have these little tiny organizations, as you can see as I've highlighted in the red and blue here, is the red is going to represent actin, and the yellow is going to represent myosin, and we're going to start talking a little bit more about that. But this actin has this ring-like structure around it, and so like right here, this little red would be like the nebulin, which we'll talk more about. And then this green, this is going to represent the tropomyosin right there. And then these little red dots, Those are going to represent something as well, which we'll talk about in a minute. Those are going to be our, oh man, why am I totally spacing the name right now? Those are going to be our troponin molecules. But we'll talk about that a little bit more in depth as we move down here. Something else important is on this myosin strand, you have these little things called myosin heads, and these are really important. These myosin heads are going to be actually allow, at least as far as our current understanding goes, is what actually allows the muscle to contract. And so these little myosin heads basically are like little ratchet straps, and they're going to just tighten the muscle. And we're going to talk about the mechanism of how all these little myosin heads actually work. But this breakdown right here is just the general understanding of, as I explain more about muscle, what exactly we're talking about, so that you don't think I'm talking about like the muscle belly or the paramecium or a fascicle or something. That we're actually talking about something much smaller that's way, way, way smaller. It's like little bundles of hairs. We're going to be talking about an actual individual hair rather than like, for example, like an entire ponytail. Might be kind of a silly way to describe it, but we're actually going to be talking about these individual little, I should actually get this out. When we talk about a muscle fiber, this, where this magic is happening, is what we're really talking about. So as you can see here, I've drawn it out previous to this just to try to make it, like I mentioned earlier, make a little bit more sense. This right here, I'll just go ahead and, oh my goodness, I don't know how that happened. But I'll just go ahead and label this right here. I made this big line pointing to this little guy right here. And this is an actual myosin head, or myosin filament. Head all blown up. And right here, I'm just going to label these as we go. These are the binding sites. For ATP and such. And we're going to talk a little bit more about that, what that means in a second. These are going to be your thin filaments. I'm just going to get this vocabulary out there right now, actually. That makes a little bit more sense in my head when I explain it. Thin filaments and thick. I'm not going to write filament because I'm lazy, but anyway, and then right here, this is really important. This is called the hinge point. And at the hinge point, and there's actually another region right here. Oh, let's go back. There's actually another hinge region right there. But these hinge regions are really important. And just remember that word, hinge region, as I explain more, because that's going to come into crucial play here in a moment. But let's move back over here. So what you'll see kind of labeled out right here is the muscle fiber at its very most basic. And that's really important because, like I mentioned earlier, this is where the magic happens. So each of these little components in this area is all this right here. That is where we're going to see the muscle actually be able to contract through something called the sliding filament theory. And so I'm going to start labeling a couple extra things. Out here, like I mentioned earlier, we have our, oh, why am I forgetting this? So this is an actin, right? Wouldn't this be all part of it? Oh, no. So like this right here and this right here, this is what we're going to call an actin thin filament. And an actin thin filament has a bunch of different components, which I'm going to label. This right here obviously is the actin. And then this area right here, this green area, this is what we call the titin. Okay, titin is like a supporting protein that helps hold the actin and the myosin and the nebulin and the tropomyosin and all those things together kind of helps maintain its structure. And then right here, this little region, which I'll just highlight, this region right there, this region is what we call the Z disc. There's these little proteins that kind of have like these circular shapes to them on each of these, which I forgot to draw earlier. But this Z disc region, again, allows the different sarcomere little protein cells to actually attach to each other, so that you have a bunch of these actin thin filaments attached to myosin, which attacks to more myosin, which attaches to more actin thin filaments, and so on. So you kind of get this row of these thin filaments. And then what else can we label here? Right here, this obviously like the big, this blue protein has these little red, actually, we'll just go over here. So this, right, I have pointed is just a little indication of what I'm talking about more over here. So we'll zoom in and kind of talk about this. So right here we have what we call troponin. And troponin is at a 1-to-1 ratio with tropomyosin, and we'll talk about that in a second. But with troponin, we have 3 different subunits, as you can see. We have the C subunit right here, we have the I subunit right here, and we have the T subunit right here. Each subunit attaches to something different. The troponin T attaches to tropomyosin, the troponin C attaches to calcium, and the troponin I am actually not sure exactly what it attaches to, so I'll have to do a little bit more research to remember that specifically, but I can't remember it off the top of my head, but I don't think that's super essential for the purpose of teaching this video right here. So each of these blue circles that I have written or have drawn, these are individual G-actin molecules. And together, an entire string of them is what we call F-actin. And this is really important because these protein structures are what allow the myosin to, these regions are what myosin binds to. So these are like the anchoring points for the myosin to actually cause muscle contraction. So that's super important. Now this green, this isn't a super great drawing, but hopefully it should get the point across. This is like a little, almost like a little coil. And this protein structure is actually what we call tropomyosin. Now, tropomyosin is really important, especially in skeletal muscle, because it allows for a lot more controlled contraction, because this tropomyosin actually blocks myosin heads from, so you have tropomyosin and myosin, they're kind of like antagonistic of each other, I guess. The tropomyosin actually blocks myosin from binding to these regions of, to these regions right here, called G-actin. So that's really important. And now you can see right here, this little like red thing that I'm tracing right here, or excuse me, this yellow that'll go away. So that yellow, oh, forgot to switch to my pen here. The yellow is actually, it's not an individual protein itself, but it's actually a region on these G-actin molecules. And these are the binding sites, or excuse me, the active sites. I guess active or binding, I don't think it really matters, because that's, the reason they're active is because that's why it's where it's binding to. These G-actin binding sites or active sites are where the myosin heads will actually bind to when tropomyosin, again, back to tropomyosin, when it actually moves out of the way. And tropomyosin is moved out of the way by calcium. And so when calcium is exposed to the cell, which comes in from the sarcoplasmic reticulum, or I guess it could also come from extracellular space as well. But when this calcium actually binds to the tropomyosin, it actually moves tropomyosin out of the way and allows these regions of the active sites to be exposed and moved out of the way so that a myosin head can actually reach up and attach to them. And obviously that's really important because as I assume you're gathering from this is where the magic happens. That's how the actual muscle contraction is able to work, is thanks to calcium moving tropomyosin out of the way, exposing the active site, allowing the myosin head to bind to that binding site and begin its process, which we'll describe as the sliding filament theory, because we're not 100% sure how it works, but we're pretty sure. And so anyway, the last important thing to mention, this is called nebulin. Nebulin is a structural protein. As you can see, the troponin, the tropomyosin, the G-actin, which forms F-actin, all binds to nebulin, which is, it's kind of like the foundation in which all these proteins can kind of bind to. So anyway, that is that. So now we'll describe something, this little diagram I have written here. This little diagram is representative of basically everything I drew right here and right here. This is just a kind of simple way of explaining the certain regions that, I guess, when we're looking at all of this like under a microscope, we need to be able to talk about certain regions. And so these proteins kind of show up differently under a microscope when you're examining them. And that's how we're able to kind of name all of these little areas to help us understand in a more rudimentary way how the structure of these proteins and muscle cells actually work. Okay. So I'll kind of point out how it relates to all this in a minute, but first let's label a couple things. This green is what we're going to call the Z line, okay? And the Z line is made up of a darker band when you look at a sarcomere under a microscope. And it's just an identifying point to kind of know, because based on experiments and obviously our understanding of muscle, we know what proteins and structures are located under the Z line. And like I said, this Z disc, so this orange area actually is the same as this Z line. This is just a more simple way of showing it. So I'm going to draw Z line over here too so that you better understand it. In the middle here, these big blue lines, these are what we call thick filaments. Thick filaments. Let me make sure I'm looking at one of my notes here to try to make sure I describe this correctly. But if I recall correctly, yeah, the myosin heads link the thick and thin filaments together, forming the cross bridge. So these thick filaments are really where we're going to see this this area of like this. This is going to be what we're going to see here is thick filaments, is where the myosin is actually binding to the F-actin and such. And so that is why they're called thick filaments, is because that's where all the myosin heads are, and it's going to appear very thick when you're doing like a cross-section of a muscle cell. Another important thing to mention is the length from each Z line to the other Z line is what we're actually going to identify as a sarcomere. So an individual muscle cell itself. Now, obviously a bunch of muscle cells are all going to be lined up next to each other in order to create what our muscle is. But this is if we were zooming in on one muscle cell, on one sarcomere, and showing how the protein is structured inside this sarcomere. And then next to the thick filaments, we're going to have thin filaments. Thin filaments. And now we're going to identify a couple regions here. I already have this one labeled. This is the H zone. Outside the H zone, this little distance just right here in between the two thin filaments is what we call the H zone. And the width of the thick filaments is what we're going to identify as the A band. So A band to here, and then A band. And then outside of the A band, we have the I band. Oh, I just realized that I had already drawn these out right there. But anyway, okay, so this, I band right here, and then A band right here, and then I band right here. Sorry, I forgot I drew those. But anyway, that's kind of how we identify the regions. Now, the A band is part of the H zone. And so that's a little bit hard to explain, but I'll do my best to draw it out over here. So let's say we have this right here, this is where our myosin heads are attaching to. I'm just going to draw a quick drawing. So next to that, we're going to have we're going to have multiple little myosin chains all stacked up next to each other, okay? And off each of these, we're going to have our little titin fibers. Okay, and then off of each titin fiber, we're going to have our little nebulin here. Or not nebulin, our little actin filaments. Obviously, nebulin's in the middle. Okay. Anyway, this is going to be a really fast drawing. And then obviously it's going to continue this way. But I don't have a lot of space. Anyway, out here on the periphery, this is where we're going to have our little Z discs. Okay, Z discs. It's going to kind of look something like that. Anyway, in between each length of Z disc, in between the Z disc, this is what we call the sarcomere. I'm using the book a little bit to help me kind of explain this so I get my drawing straight. And then in this area, the H zone is, so let me have the H zone, is also going to be from Z disc to Z disc. And then in the middle, in this, basically just focusing on this region that surrounds the actin proteins with the myosin heads and myosin filaments and all that, this is what we're going to call the M line. M line. And that literally just means middle line. And that's just important because there is a center to each of these actin, these F-actin filaments that the troponin and all that they're binding to, that they're basically pulling the F-actin into the center of the why am I literally forgetting everything I know right now? Of the myosin. So anyway, that's how we get the midline. Anyway, I think I've got everything so far for this. So let's move down a little bit and start explaining how this all actually works. So, like I mentioned earlier, out here, around each muscle fiber, so this is where we're focusing now. Okay, so each muscle fiber has, basically what we have is a little area for, you have a muscle cell. So I'll actually draw this like this. Have an actual muscle cell. And around the muscle cell, we have the sarcoplasmic reticulum. And it's just kind of this mesh area where around each cell sarcoplasmic reticulum. This is where the calcium is stored. And on either side of each of the sarcoplasmic reticulum, kind of in like a little zone like this, is what we call a T-tubule. And these little T-tubules are just invaginations in the membrane that allow for action potentials to take place, because they're going to be little spots for calcium and potassium to flow in and out of the cell. And I'm going to explain that a little bit more as we go. Now, this area, these three together, so obviously like out here we would have another sarcoplasmic reticulum and so forth. But this is what we call a triad. And that's pretty an important term, just because it's referring to two sections of sarcoplasmic reticulum and a T-tubule, and that's going to put together our little muscle cell. And then in between the T-tubule and the sarcoplasmic reticulum is DHP and ryanodine receptors. And these are just special voltage-gated sensors that allow action potentials to travel through the cell and create, dihydropyridine, and ryanodine receptor tangles, Anyway, now we're going to kind of zoom in a little bit more on that. And so these DHP and ryanodine receptors are going to play a very key role in how we actually get action potentials to go through the cell. And so I've drawn out a little diagram right here. Okay, this is sarcoplasmic reticulum again. This is our triad again, like I mentioned. And so this is where we're going to get our action potential. So the first thing that's going to happen is we're going to get an action potential. So I'll just draw action. Actually, I'll do it in yellow. Action potential down this is a neuron, by the way. And what's going to happen is we're going to get an action potential, a little boing, that comes down the neuron. And what this is going to do is this is going to stimulate calcium. So let's change this to calcium. It's going to stimulate calcium, and these little snare proteins are going to bind to the little vesicles full of calcium. Going to bind like this. And calcium is going to go through from the neuron through the synaptic cleft, right, which is this area right here, and is going to bind with these snare proteins, and when the calcium binds to these snare proteins, what we're going to have is the release of acetylcholine. Sorry, I explained that a moment ago. Now, when the acetylcholine binds to these, when it's released from these vesicles and is released into this little region right here, the acetylcholine is going to leak out and open these ligand-gated potassium and sodium channels. And what that's going to do is sodium is going to rush into the cell. Okay, sodium is going to rush into the cell because it has a higher driving force than potassium. And anyway, we're going to see this sodium rush into the cell out of the sarcoplasmic reticulum, and it can even enter all the way over here through these little gates and go into the T-tubule. And then in the meantime, what's going to happen is the acetylcholine is going to get broken down by acetylcholine esterase. It's going to break down the acetylcholine moving into the synaptic cleft and prevent that signal from continuing to travel farther. And then what's going to happen is as this BWOG, so now we've got this BWOG moving into the cell. As it is flipping charges and that length constant is moving down the cell, what's going to start happening is these channels in the T-tubules called DHP receptors, dihydropyridine receptors, it is going to release the signal. So when these are activated, it's actually going to release calcium from inside the sarcoplasmic reticulum to relieve the sarcoplasmic reticulum. And it's going to leave the sarcoplasmic reticulum, and what it's going to do is it's going to move into the cell, and now the calcium can leave, and what's going to happen is the calcium, in a roundabout way, is going to start binding to this tropomyosin. And what happens is when this tropomyosin binds to the active sites, it actually moves tropomyosin out of the way. Okay, we're going to get tropomyosin to move out of the way. It's going to go more like this now. And now these binding sites are going to be exposed. And now the actin filament can actually bind to the active sites on the actin, because this calcium has moved the tropomyosin out of the way and allowed these myosin heads to bind to the active sites. And now when tropomyosin has the ADP and this phosphate attached to it, it's what we call a cocked state. And that just means it has a high energy, kind of like when you pull the trigger back on a gun. And this high energy state obviously wants to release that energy. And what's going to happen is as it binds to the as it binds to these active sites and starts forming these cross bridges, it's going to go through a conformational change that moves tropomyosin off of the active site. And when it binds, the rest of the energy, so like it's going to bind and it's going to want to kick these two molecules off of it, and it's going to release heat. And when it releases heat, it's going to go through another conformational change, kicking off this phosphate and this this ADP and this inorganic phosphate, and it's going to bend forward, as you can see with this arrow here. And that's actually going to move this entire myosin chain. And then what's going to happen is a little ATP is going to come along, and it's going to bind. It's going to bind right here to the actin. Pull the actin past the myosin. Yeah, so it's going to bind to the myosin, and it's going to release it. See how there's a gap here? It's going to release it from its binding site on the actin. So when that ATP binds to it, it's going to pull it off of the actin binding site, and it's also going to give it energy again. And that's why you can see right here is this G-actin, this angle right here, or excuse me, this myosin has a much bigger angle in between it. And that's because now with this energy, because of the ATP, it now has a lot more energy. And it's going to keep continuing that process of binding to the actin site, hydrolyzing the ATP into ADP and inorganic phosphate. It's going to release heat, which is going to allow the myosin head to curl forward, sliding the filaments past each other, and then the ATP is going to come along, bind to it, give it more energy. The myosin head is going to hydrolyze it, turn it into ADP and inorganic phosphate, and then it's going to return to its original cocked position. Now, obviously, this is a pretty long video here, but there's a few different ways that the muscle can actually receive the energy necessary to go through this Crossberg cycling. And that is through a few different types of energy. So the first type of energy that we have to even be able to allow this entire process to happen is because of systolic ATP. Okay, systolic ATP is the number one and most important form of energy that is utilized in the muscles. Now, systolic ATP is just what we call, it's kind of in the term. When we hear systolic, it's because it's in the cytoplasm of the cell. It's just present in that cytoplasm. The cell, all the cells have it as energy, especially muscle cells. Obviously, all or the majority of cells need ATP to function. But with systolic ATP, what you have is the most immediate form of energy for the cell to use. It does not use oxygen, which is nice because I'll just say no oxygen. It does not use oxygen, which means it's a very readily available form of energy, which means it's already there. Now, it provides high energy, but it's short-lived, which means although it will provide a burst of energy to allow muscle contraction to happen, it doesn't mean it's going to last forever. After we run out of systolic ATP, we're going to move down to creatine phosphate. Creatine phosphate is another form of energy that the cell can use once this systolic ATP has ran out. And it is another rapid energy source. Now, basically what creatine phosphate does is it basically readily attaches ADP and inorganic phosphate to the myosin heads, allowing them to continue to do their job without requiring oxygen as well. No oxygen. Let's see. This type of energy does require a special enzyme. Requires, I know I spelled that wrong, by the way. Requires enzyme called creatine kinase to actually get the creatine phosphate into ADP and inorganic phosphate in order for the cell to use it. However, it is usually when you take creatine as a supplement, for example, there's enough there to basically replenish all your stores as you need. but it's not as useful as systolic ATP. The next, glycolysis. You get all your inorganic all your ATP from the breakdown of glucose. So as glycolysis says in the name, this is just the breakdown of glucose into ATP, which it is very high in ATP. However, the glycogen that the glucose comes from is stored in the muscles, and it can also function with no oxygen. So usually when you're shooting for something like hypertrophy while working out, after your fatigue, like the fatigue in your muscle starts to set down, you usually burn through your systolic ATP and then your creatine phosphate, and then your muscles begins to actually deplete the glycogen stores in the muscles, which is, great for you, obviously. Muscle lifting is important. However, weightlifting is important for your muscles. But this glycolysis is not very fast. And so it says right here that although glycolysis can be good support for intense muscular activity, it can only be sustained for a minute before these muscles actually begin to fatigue. And then lastly, we have aerobic or oxidative respiration. This can be sustained for about a minute as well. Let's see, blah, blah, blah. Okay, so this does require oxygen. This does require oxygen, and this energy source comes from the mitochondria. Now, this is a pretty slow form of energy compared to the previous three. Anyway, and it's the fourth type of energy that your muscles can use in order to produce muscle contraction. Now, there's a few different types of skeletal muscle. Man, I still have a long way to go. I got to get all the way through smooth muscle still. Okay, these are the types of skeletal muscle. Each type of skeletal muscle uses a different type of energy primarily before the others, and so we'll kind of talk about the differences in between each type of skeletal muscle. So to start with slow twitch oxidative, you have a slow, as it intends with the name, you have a slow myosin ATPase activity. These are usually smaller muscles. Let me make sure I didn't forget anything in the previous. I need to edit that part out just because I was regathering my thoughts. These can actually have long contraction periods. They are fatigue resistant. They use mostly aerobic. The aerobic type of energy, and they contain high numbers of mitochondria, and a high blood supply. The fast twitch oxidative muscles, you're going to have, let's see, you can have a few types here. This can be considered 2C, 2AC, or type 2A. Now, they have fast ATPase activity. These are a little bit larger in size compared to the slow twitch muscles. These are a little bit more fatigue resistant than the next type. Fatigue resistant. These are also going to be a little bit more red, which means they have myoglobin. And then lastly, we're going to have our fast twitch glycolytic muscles, which is going to be our most fatiguing muscles, but also our most powerful and most energy consuming. So these are going to be larger muscles. They are going to be fast with their circupump activity. The other two were a little bit slower. And it can use multiple energy types. And it is white, which means it has low myoglobin. Low myoglobin. Anyway, those are the three types of skeletal muscle. Next, we're going to talk about smooth muscle. Okay. Now, I have everything drawn in here, and then what we're going to do is talk about how smooth muscle differs from skeletal muscle. So smooth muscle is unique because it has an ability to hold contractions for a very long time. And because a lot of the time your body just does it... Man, I swore my do not disturb was on. It will hold contractions for a long time. This is things like peristalsis. Or controlling your vasodilation. Sphincters. And things like that. So first we're going to talk about specifically phasic. Phasic contraction is interesting because what you have here is you have an unbound myosin head, okay? And this unbound myosin head cannot latch on to... this myosin head cannot bind to the actin unless it has its phosphate and its ADP. Okay, and so what's going to happen is we're going to have calcium right here come into the area, and we're going to have MLCK, which is active because of... let's see, ADP, ATP, MLCK. Okay, yeah, so I was just making sure that I had all my stuff right. So something called calmodulin, which is kind of interesting. Anyway, it has this little binding site right here. And normally this tail goes right here, but because of calcium, calcium is going to come in and unbind calmodulin from itself, which is going to be able to make MLCK active. And when MLCK is active, hidden active site, yes. So what myosin, what the MLCK is going to do, is going to phosphorylate the regular light chain of the myosin molecule, and it's going to allow this this myosin net to actually move up and actually bind with the actin binding site, thanks to MLCK. And then what's going to happen is once phosphorylated, the MLCK will attach to the actin and release a phosphate, as you can see here. It's going to be an extra phosphate, and it's going to bind to the myosin protein, and it's going to stay in this cocked position. So anyway, this extra phosphate right here is going to attach and allow it to stay in this position here. And when the heat is released, as you can see here, and the ADP and the phosphate are removed, it's going to propel this thing forward. And when it propels forward, it's actually going to move. It's actually going to move this. This represents the actin, by the way. It's actually going to move the actin towards the direction in which the myosin had pulled. And then what's going to happen is this extra phosphate is going to, this extra phosphate right here, and I'm forgetting what I'm supposed to be teaching here. This extra phosphate is going to bind, and then it's going to release heat, this extra phosphate and the ADP, and it's going to push the myosin forward. And then we're going to get more ATP in here, and still you're going to have this second phosphate bound. And so if we don't have MLCP, and to cleave off this phosphate, what's going to happen is it's just going to skip this step, and it's just going to move here, and it's just going to basically essentially stay contracted. However, if we do have Rho kinase, which is going to be what activates MLCP, if it is not activated, which does not cleave the phosphatase. So the Rho kinase is going to inactivate the phosphatase. And when it's inactive, it does not cleave off this phosphate, which allows the phasic to stay just in this little, basically in this little bottom stage here. So it just keeps moving like this. And it just stays in that circle. However, if Rho kinase does get activated, what it does is it actually allows phosphatase to move over here and cleave off this phosphate. And then what's going to happen is it's going to follow this path. And calcium is actually the one that activates Rho kinase, which is funny enough. So Rho kinase actually turns on MLCP. So that's only if more calcium gets exposed in or admitted into the cell through the sarcoplasmic reticulum of smooth muscle. And then this stage right here, if it is active, this is what we call the relaxation phase. As you can tell, it is not attached. And so you just have the phosphate and the ADP waiting for calmodulin and MLCK, which this is calmodulin, by the way, and MLCK to come through and bind to this and begin this cycle. Now, that is what we call phasic contraction. Now, tonic contraction is a little bit different. So tonic is a little bit different. We have the similar counter, or we have like similar components. We have calcium, MLCK, and ADP. However, what's different is anyway, tonic is going to use something a little bit different where tonic actually does not require ATP to stay latched. So once MLCK is active because of calcium, it attaches to the myosin head, which obviously releases phosphate, releases ADP, releases the MLCP, and releases the other phosphate. And that release is what cocks this forward and allows this movement head forward. Now, if there is not enough MLCP, then MLCP will not be able to cleave off the extra phosphate, which just keeps this thing latched like this. And so as you can see right here, it's unlatched. So actually this latch phase should actually be more down here. But it's going to latch and it's going to stay latched until hydrolysis occurs, as you can see right here. And hydrolysis is going to just turn the ATP into ADP and phosphate, and this will result in the restoration of the higher energy phase, which is what you see right here with the ADP and the inorganic phosphate. So this would be anyway, this would be the lower energy form. Oh yeah, here's a little example of MLCK. Anyway, now we have reflexes. So reflexes are going to occur, instead of going all the way up to the brain, they're going to just occur in the spinal cord. And I'm going to talk about kind of how all that works. So basically in the brain, you have a gamma motor neuron, which is going to attach to the muscle or an intrafusal fiber. And these sensory fibers, as you can see right here, we have type IA, II, and IB, all have different jobs. They're either going to sense the type of tension that is being released, the rate of tension, and things of that nature. And so inside each muscle, we have two different types of fibers. We have a spindle fiber and a bag fiber, as you can see, like right here. This is the. Actually, let's just go back. Get my highlighter. Oops, it's way too thick. Right here we have the bag fiber, the spindle fiber. And right here we have the bag fiber. And that's going to be inside all of our muscles. And each fiber has a different job. So for example, our chain fiber has the job to basically detect the rate at which stretch is happening. And the bag fibers are going to detect if stretch is happening. And so these stretch fibers are actually full of fluid, and they're surrounded by different neurons. For example, the spindle or the chain fibers are surrounded by a type 2 alpha motor or gamma motor neuron. And the bag fibers are surrounded by a type 1A motor neuron. Essentially, what's happening is anyway, essentially what's happening is these fibers are just detecting when your muscles are moving to help with proprioception. So for example, if I'm curling my arm, my tricep is going to begin to be stretched. And when my tricep is stretched, the bag fibers are going to pick up on the rate at which that's being stretched. And so basically it's just communicating to my brain what is happening all over my body. Next, we have something called the Golgi tendon reflex. And the Golgi tendon reflex essentially happens when you feel a muscle begin to be stretched. So right here, for example, in this little area, let's say we have our quadricep. And our quadricep is going to attach below our knee in this region. And normally the tendon doesn't stretch. But when the tendon begins to be stretched, it's going to send a signal back to your spine that says, hey, I'm being stretched too much. We need to stop contracting this muscle because we're going to hurt ourselves. And so it synapses in the spine. It actually never goes up to the brain. It actually just synapses in the spine and then sends a signal back here to begin contracting the hamstring, basically to do the opposite motion of the leg. So it's going to shut off contraction here and it's going to start contraction there to keep the muscle from being stretched too much. For example, like I mentioned earlier with the quadricep, if you were lifting a ton of weight. And so when it senses the excessive tendon, when it senses the excessive tension, it's going to tell the quadriceps to inactivate and tell the hamstrings to begin to activate. And that's just going to be, the whole point of that is just keeping you safe. And that's just called the Golgi tendon reflex. Then the cross body reflex or the cross extensor reflex is essentially the same. So for example, if I was walking with my right foot and I stepped on something, and I stepped on something right there, and it's super sharp. Let's say that's my right leg. What's going to happen is instead of pushing down like I was originally with my quadricep, it's going to unactivate my quadricep, start activating my hamstring to help get my leg out of the way. And at the same time, it's going to synapse and send another signal to the other side of my body and tell the quadricep on my right side to extend to help support my body in order to get weight off of my foot so I'm no longer stepping on that sharp object. And that all just synapses in the spinal cord, never even reaches the brain, and it happens almost momentarily. Anyway, I think that should be everything for the video today. Thank you for tuning in.